Thank you. Thank you for the organizer to invite me. I have a special mention for Manfred. I thought he was a friend before he asked me this talk, but so molecular difference, the title is Molecular Difference Between Colon and Rectal Cancer, and in fact, I don't know, but there is few data in the literature on that. In fact, there is three major phenotypes if we consider colon cancer. Let's say colorectal cancer. There is SIN phenotype, that means chromosomal instability phenotype. There is MSI phenotype, microsatellite instability phenotype. And the third phenotype is a SIMP phenotype, which means that hypermethylation phenotype. What we know is that the distribution of these different phenotypes are clearly different from proximal to distal part of the colon. MSI and SIM phenotype are more frequent in proximal than in the distal cancers, but there is few data concerning the comparison between colon cancer and rectal cancer. This is probably the most important series in the literature. There is, as you can see, approximately 700 proximal cancer, colon cancer, 700 distal colon cancer, and approximately 1,000 rectal cancer. The first part of this slide concerns the MSI phenotype. That means deficient mismatch repair genes. That's the MSI phenotype. And the proficient mismatch repair tumors, that means the stable tumor. So what we can see, and everybody known in this room, that MSI tumors are located in the proximal part of the colon. And as you can see, there is no difference or very few difference between distal and rectal cancer in terms of prevalence of MSI phenotype. Just to mention, most of these patients here in red, the MSI proximal part of colon tumors is in fact sporadic MSI linked to a hypermethylation of MLH1 gene. And these patients in red, in distal and rectal, the vast majority of these patients are HNPCC patients. Now, if we consider the SIM phenotype, as you can see, here you have approximately 40% of the proximal colon cancer with this hypermethylation phenotype in the proximal colon cancer, and you have quite the same percentage, the same prevalence, approximately 10% of the distal colon tumor with a SIM phenotype, and it's approximately the same percentage for rectal cancer. So there is no difference between distal and rectal cancer in terms of prevalence of MSI or SIM phenotype. Now, if we group now the four categories, the two categories, SIM phenotype and mismatch repair, you have four different groups of tumors. And once again, there is a difference between proximal and distal and rectal cancer. What we observed here is the sporadic MSI tumor in red. That means patients with hypermethylation profile with MSI tumors. Here we have the SIMP phenotype with a proficient mismatch repair system tumors in blue with approximately 20%. And here we have specific deficient mismatch repair without hypermethylation phenotype, and these patients are clearly the HNPCC patient. So you have the same, quite the same percentage, and it's probably very correlated with the surface of mucosa in terms of occurrence of HNPCC tumors, since you have less HNPCC tumors in rectal part of in the rectal part, just because the surface of the mucosa is much less than in the proximal part and the distal part of the colon. So in fact, all the difference we observed is the difference between proximal and distal part of the colon, and not between colon and rectal. This is the same thing for P53, in blue P53 mutated tumors, and in red P53 non-mutated tumors. There is no significant difference between the frequency of the occurrence of mutation, and this is quite the same for KRAS mutation. Maybe there is more KRAS mutation in the proximal part than in the distal or in the rectal part of the colon. Now, if we go deeper and try to see a difference in terms of copy number variation, and if we compare, and this is a series of rectal cancer, approximately 100 rectal cancer. So in green, you have the gain, a gain of chromosome, and in red, the loss of a chromosome. And if you go to a very frequent alteration, what you can observe in rectal cancer is the loss of the short arm of chromosome 17, the loss of the chromosome 18, and also the loss of the short arm of the chromosome 8, which are, in fact, the most frequent alteration found in colon cancer, and it's exactly the same profile. What you can also observe is a gain of chromosome 20, the gain of the long arm of chromosome 13, which are, once again, the most frequent alteration that occur in colon cancer. So there is no difference in terms of copy number variation between colon and rectal cancer. So since there is no data in the literature concerning mutation in the different pathway, I looked for what the data are available in TCGA, the Cancer Genome Atlas initiative, which put in the online all what have been done in terms of exome sequencing in different tumor types. And in particular, there is approximately more than 250 colon and rectal tumors that have been sequenced in the term of exome sequencing. So I extracted from this database only what occur from stable tumor, because if I include MSI tumor, I will find differences, but these differences is due to the proximal colon and due to the microsatellite instability and the deficiency in mismatch repair system. So what I present here is only the stable tumors without a deficiency in the mismatch repair tumor and without the hypermutable phenotype. So and this is a Wnt signaling pathway, which is one of the most activated pathway in colon cancer. And you have here, for example, APC gene, which is represented there. And you have in the, this is a box of APC, and you have here the frequency of APC mutation in colon. And you have here in this part the frequency of mutation in the rectal. So what you can observe, and in blue is inactivating mutation, in red is activating mutation or amplification. And what you can observe, for example, for APC, you have 70% of mutated tumors in colon, and you have 85% of the rectal tumor that have APC mutation. For example, for beta-catenin, you have 5% in both cases of activating mutation. Now, if you pull together in terms, if you integrate all this data in terms of pathway, what you observed is that you have an activation of the Wnt signaling pathway in 87% of the cases in colon cancer and 95% in the rectal tumors. So there is no significant difference in terms of activation of this pathway in terms of mutation. Now, if we go to the TGF-beta signaling, which is also a pathway that are inactivating in a colon cancer, what we observed is a very similar rate of mutation in the different genes that participate to this pathway. And once again, 28% of the colon cancer is have an inactivating of this pathway as compared to 29% in rectal cancer. And this is, for example, for SMAD4 genes, which is one of the most favorite gene of Arnaud. There is exactly the same percentage of mutation between the two types of tumors. So if we integrate the Wnt signaling pathway and the TGF-beta pathway, which are related by their influence in the MYC genes, there is exactly the same prevalence between colon and rectal cancer. Now we can go to the PI3 kinase mutation, which has been described by Gunnar just before. Once again, the prevalence of inactivating mutation in PTEN, which is an inhibitor of this pathway, there is exactly the same prevalence. It is the same for PI3 kinase AR1 genes, and also for the frequency, the prevalence of PI3 kinase mutation in the two types of tumors. And once again, there is exactly the same prevalence in terms of activation of this pathway in colon and rectal cancer. We can also integrate the, let's say, the EGFR pathway or the MAP kinase pathway. And as you can see, once again, the prevalence of activating mutation in NRAS is very similar. Maybe there is an increased mutation rate in KRAS, in rectal cancer, as compared to colon cancer. But if we integrate all these stuff together, we have, let's say, the same percentage, a non-significant difference between the rate of activating of the MAP kinase pathway. So now if we summarize these two types of pathway, in the receptor tyrosine kinase signaling pathway, we have the same activation of these two pathways. Now we can also speak about the P53 pathway, which is, let's say, there is few genes that are instead of P53, we can also integrate ATM genes, and as you can see, there is exactly the same prevalence in terms of mutation. Now, if we go, and I finish by that, in the difference in terms of gene expression between colon and rectal cancer, there is very few data on the literature that compare directly rectal and colon tumors. Here is a publication that, let's say, studied all the gene expression by Affymetric chip array. So you have the right colon, the left colon, and the rectum. They considered, in fact, the genes that are differentially expressed between these three different types of tumors. So between right colon and left colon, there is 40 genes that are differentially expressed, significantly differentially expressed. And as you can see, there is 50 genes that are differentially expressed between right colon and rectal tumors. There is no genes that are differentially expressed between distal colon cancer and rectal cancer. And if we put together right colon and left colon and consider colon and compare to rectal cancer, there is only 60 genes that are differentially expressed. So this is a list of genes. So I have no clear conclusion to give to this table. Maybe you can find your favorite genes, but for me, there is no clear explanation what are behind this list of genes. As you can see, there is mainly transcription factor that are differentially expressed between colon and rectal tumors. So in conclusion, I would like to say that there is no evidence that rectal cancer are different from colon cancer. There is evidence that rectal cancer are closer to distal colon cancer than distal colon cancer to proximal cancer from a molecular point of view. And the main difference we observed is in fact due to the phenotype of the tumor and not due to the tumor locations. And if we consider non-hypermutatable tumor or MSS tumor, there is no evidence of any difference between rectal and colon cancer in terms of molecular differences. Thank you.